So I'm very excited to start with the first speaker, Professor Jackie Roth. She will be speaking about mainly assay validation, clinical regulatory laboratory regulations regarding assay validation, and she'll bring an example to the actual validation of these NGS-based sequencing data in the lab. So thank you very much, Professor Roth, for being here, and we are very excited to hear your talk.
Thank you so much, Dr. Miho. Are you able to hear me okay and see the slides?
Yes, all perfect.
Fantastic. First, I would just like to thank Susanna and Ulrich for the invitation to come here. I'm very excited to speak to all of you today about the basics of assay validation and what we do to bring our assays into the clinical genomics laboratory. I'll apologize in advance. Some construction is going on outside that I cannot control. So if there's a little bit of background noise, my apologies. Today, we'll be talking about 3 different parts. So learning when an assay is ready for clinical implementation. Once you've made that decision, how to validate and verify an assay. And I'm going to be speaking mostly about the US regulations in terms of what you would need to do to make this happen. And then last, I'm very excited to share about our participation in a cooperative group validation that we did for an NGS oncology assay. However, the principles that we've learned from that validation could very easily be applied to your community and T-cell and B-cell receptor gene sequencing rearrangement. So with that, I think we're all familiar with the process of translating new discoveries into clinical care. There's a lot of research going on that's aimed to discover different genes and proteins that might be important in disease, with, of course, the goal of translating this into clinical care. Once we're able to go through that process, we can implement certain assays for clinical use, and they can help to be used to diagnose, decide prognosis and treatment decisions for our patients, right? So that's usually the goal of our research, is to get this into some form that can help our patients. But there are many steps along the way, and this, of course, can take not only months, but years for some assays. This is the example of a laboratory test development process that we follow at Penn, and I'm going to start going through this at the top here and discuss how we decide if a test is ready for implementation in the clinical laboratory. In our test development process, there are actually 2 different ways that we receive requests. They can come either internally from within our division, or perhaps an external request from somebody outside. So internal requests to launch an assay clinically arise from a variety of different ways, including reviewing tests that we currently send out. So maybe your institution is sending out all of their T-cell receptor rearrangements, and you want to bring it in because it's costing a lot of money. There are certainly pandemic situations that dictate we develop a lab test ASAP, as we've all just seen, as well as literature review and seeking to improve other current tests. We also receive tests from external members. These could be clinicians, these could be researchers, they could be anybody in our community. And we've developed a formal request pathway, including our submission form, committee review, and an official response. I've included the link, and this arrow here is pointing to our Word document that shows you our external test request form. It's publicly available, so you all can take a look. And there's about 5 pages of questions in which a requester must really provide the information that their assay is in fact ready for the clinic. So whether we receive a request internally or externally, we come to a critical decision point about whether or not we will bring this assay in. In our laboratory, we discuss the following 3 criteria to make this decision. So clinical utility, which we could argue is probably the most important. We need to know that this test will aid in the diagnosis, prognosis, or treatment of patients. And maybe there's a new guideline out saying that all patients with X disease must have this test. We are, of course, a business, as these laboratories are, and we need to make sure that a test would be economically feasible for us as well as the patients. And to do this, you can even perform a return on investment calculation to determine, when you could make the costs back here for spending the money to bring on the test. And lastly, we look at institutional priority. Maybe your institution this year is focused on molecular diagnostics and infectious diseases, and therefore is not providing support or tools for immune receptor rearrangement sequencing this year, right? We've encountered things like that. So you have to have the support of the institution in order to make these happen. After this decision point to move forward, I'll now get into talking about what we do in order to verify or validate an assay in the lab. And that's at this point here in our lab test development process. In the United States, laboratory regulations actually dictate what we need to do and how we can validate this clinical laboratory assay. So if we speak of the bare minimum regulations in the United States, that is going to be the Clinical Laboratory Improvement Amendment of 1988, which was passed by the US Congress to ensure the basically the minimum quality of clinical laboratory testing. So this is a certificate that you might see United States labs have as being CLIA certified. CLIA oversight is carried out by a variety of different agencies, and the ones you'll hear me mention are CMS, or the Centers for Medicare and Medicaid Services, that's going to publish the regulations that will be important to us validating the assays, as well as the FDA, the Food and Drug Administration, which is going to be looking at our complexity categorization. And again, that will dictate how you validate or verify your assay. Above and beyond CLIA, we have different organizations in the United States that actually have deemed status from CMS to accredit the laboratories. If I speak of our laboratories at Penn Medicine, we are accredited by the College of American Pathologists, which has regulations that are either equal or more stringent than CLIA. So by following the rules and regulations of CAP for our validation, we've automatically met the CLIA rules and regulations. Interestingly, one step even further beyond that, there are some states with some very extreme regulations. Examples are Washington and New York, where their state regulations are so extreme that they automatically get CLIA exempt if they are state certified. As I mentioned, the FDA is going to be really important in designating how we can validate these tests, as well as which category they fall into, which again will dictate the steps you need to take. So there are 3 pathways for implementing a clinical assay in the US. If your test falls into something called the waived category, these are tests with the lowest level of complexity, and the likelihood of having an erroneous result, while it's possible, it would be negligible effect on your patient. Think about these tests as being perhaps point of care tests and tests that can be run by non-medical laboratory professionals, perhaps something that you have at your primary care physician's office. On the other side of that are the FDA approved and cleared assays. And these are assays that the FDA has declared are sufficient for clinical implementation. They can be approved as the first of their kind on the market or cleared as substantially equivalent to a current test. When you do a verification, which is what these tests are going to get, and I'll go into those details on the next slide, you actually get to do less work than if you were going to make your own test. So a lot of laboratories will choose an FDA approved or cleared assay when possible. In the middle here are the laboratory developed tests, and these are the bulk of tests, at least that you are going to see in clinical labs in the United States. These are designed and manufactured and used within a single laboratory, meaning we can't become a manufacturer or company and sell our tests. That's not allowed. We assume the full responsibility for validating these tests. And interestingly, If you change even one small thing from the FDA-approved package insert on a test, so let's say it requires incubation at 95 degrees and you can only do 90 degrees in your lab, but you know the test works. Since you made that modification, you now become a laboratory developed test, and you must do the extra validation parameters that I'll tell you about. So here are the characteristics that are going to be the bare minimum for either validating or verifying your test according to the CAP and CLIA regulations in the United States. You must test accuracy and determine that your test is equivalent to a gold standard test that is out there. So you would essentially obtain either samples or reference materials that are positive for what you are looking for, such as X IgH rearrangement or certain T-cell gamma rearrangements, let's say. They have their accepted answers or known truth, and you must test that your assay can detect it in just the same manner, if not better. Precision is another metric that you need to review and test. This is going to be determining that you have the closeness and agreement of the result when you test a sample many, many times. Here, you need to do both inter and intra assay precision. You're going to run the same sample on different days. You'll run it multiple times on the same assay and ensure that you always get the same result. You'll use different technologists, maybe different pieces of equipment. Throw anything you can at it to determine that you have a high level of precision. The reportable range is a metric you must look at for both validation and verification. And this is going to be the range of values over which the relationship between your assay or your instrument or kit to which your response is valid. So an example that I can think of that might apply to the receptor gene rearrangement sequencing is, let's say that you've shown you're able to detect in a given sample anywhere between 1 and 5 different rearranged clones. That's going to be your reportable range of what you are able to detect there. You could also think about reportable range as perhaps the size of the clone or the length of the genetic sequence involved in the rearrangement. You can detect rearrangements that are 20 base pairs to 650 base pairs long. I'm definitely just making this up on the spot, but that's what your reportable ranges are going to be. You need to know where your assay is valid. And the last parameter that's going to apply to both validation and verification is, in fact, the reference range. This is the range of test values expected for a designated population of individuals, and this actually refers to individuals who are disease-free. So in addition to testing samples that are known to have the rearrangements or disease you're looking for, you must test a bunch of folks that do not have those diseases and show that your assay results as negative or not detected in those situations. You want to make sure that you would not have false positive results there. The next parameter is something that you have to do in a validation only when you are looking at developing a laboratory developed test. This parameter of analytic sensitivity, you actually do not need to redo or even verify for an FDA approved or cleared test. You basically can just accept what the FDA has shown and put out there as the analytical sensitivity, and that will be the sensitivity of your test. Namely, I'm referring to analytic sensitivity, not clinical sensitivity, which is a different metric. For analytic sensitivity, this is the smallest amount of substance that can be accurately measured by your assay. So let's say you have a receptor rearrangement that represents 0.5% of the sample that was submitted to the lab, and you can reliably detect that clone after you test it multiple times. That would be your analytic sensitivity. Your analytic specificity is the ability of an assay to measure one particular analyte or substance rather than the others. And an example of this would be you would need to show that your assay would test, if it was designed for TCR gamma, let's say, you'd need to show that you are detecting only TCR gamma rearrangements and not TCR beta. You need to make sure your assay is specific for what you've designed it for. You can throw in interfering substances in here to show that they don't cause a problem. You can do bioinformatic or in silico mapping of primers to show they won't bind in other areas of the genome. Many different ways to test analytic specificity. Much like the analytic sensitivity, the specificity is also only needed to be done if you are doing a laboratory developed test. So right here, this is a great take-home slide of the bare minimum parameters that you would need to validate or verify. Now, depending on your assay and assay design, there are going to be some additional parameters that you may need to look at for different parts of your assay. I've included this amazing table from a 2009 publication that was actually written by one of my colleagues, and I think it is an excellent development paper for just the basics of assay validation. I've included the reference at the end as well for you. An example of an additional parameter that I could think of folks may be familiar with on this call would be something like linearity that is going to apply to your quantitative tests. An example where we had to validate and verify linearity in our lab is in detecting the amount of BCR-ABL1 gene fusion rearrangements in our molecular lab in blood and bone marrow samples. We want to make sure that as the analyte increases in the patient sample, that we are proportionally detecting it accurately there. So you will determine that your test is linear over a certain given range. So I've talked a lot about validation and verification and what you do when you are at that point and you are ready to bring your assay in clinically. But it's not really that simple, especially when you're talking about a laboratory developed test. Again, different from the FDA. And before the LDT experiments, the actual validation experiments begin, we often spend months or even years or days, if you're talking COVID-19, I guess, doing different development work and testing components of our system. We'll test, do you like this Taq polymerase or this one? We'll test assay design elements. Do you like these primers? Do they work better? Or does this primer set for gene rearrangements work better? You're going to want to push the assay to extremes. Maybe what if you put in a sample that's 18 days old? Will it still work? You need to start pushing that assay in your development phase. You're going to want to establish the optimal workflow here during this. Do you do step A, then B, or B, then A? Once you've figured all of those things out, you can then have an idea of the exact experiments you would need to do in the validation with the test designed exactly as you intend to clinically implement it. As you might imagine, this can be a lot of work. So what if there was a community, such as the one you have or other ones that we participate in, where you could do this development work together? Importantly, just to be very clear, and I'll bring it up again at the end, to implement an assay clinically, the validation or verification experiments need to be done in that clinical laboratory and not in a different one. You have to replicate those experiments where the assay is going to be performed. So leading into the last part of my talk here, I'm going to bring to you an example of a cooperative group validation that Penn participated in. So about 4 years ago, we joined the Genomics Organization for Academic Laboratories that was actually founded by 2 of our former trainees. This is an academic consortium that has developed a not-for-profit structure, and it has 29 different academic participating sites. It is a partnership with the Association of Pathology Chairs here. And there are, in fact, some industry sponsors. You can take a look at that on their website. There are so many benefits to participating laboratories in the goal organization, and much of these, I can imagine, are already shared by your community. We were able to crowdsource a shared NGS assay design, and I'll get more into those details in a little bit, including bioinformatic pipelines that were able to be group designed. We have real-time development and validation support, right? Our colleagues are bringing on the same assay, so we can just call them with a question. We were able to participate in a multi-site concordance evaluation. I have the reference for that at the end if you'd like to read about it. This might be the best selling feature for some of you, especially on the business end. We participated in shared reagent purchases. Who wants to pay the top dollar when you can save some money, right? We have our real-time shared communication platforms for crowdsourced troubleshooting. Every day we're back and forth on emails. We have monthly open discussions called the Goal Cafe. Many of us take part in specialized working groups within the organization. There's availability for funding from a bunch of their academic or industry sponsors. And we can participate in practice surveys. We're helping to shape the future of NGS oncology testing by all of these institutions coming together. And you, again, will see some of that in the references at the end. So what did Penn do as a participant of GOAL, and how did we use this? Well, we jumped on board to save money right away, and we ordered reagents with the first group purchase, and we saved 90% or more. This is a huge savings when you're thinking about next generation sequencing assay reagents that are going to cost you into the hundreds of thousands of dollars. We were able to do bulk probe purchasing. This is a capture NGS assay, and save those many hundreds of thousands of dollars. If anyone is interested in exactly what we ordered, I've included a table from the publication where there were about 2,600 genes, as well as some other probes to detect a variety of other important things in oncology that we wanted to look at. We also participated in a discussion where several sites convened to identify a core gene set that we recommended everybody offer. And this core gene set, of course, was driven by current therapeutics, diagnostic diseases that needed to have a genetic component to their diagnosis, et cetera. And this is very important because what this enables is not only that we can share results and compare, but if a patient gets tested at my institution and then at yours, they might be offered the same test with the same set of genes there. So if we wanted to translate that into your community, let's say if everyone were to come up with an immune receptor test, it may be something along the lines of everybody is to offer T cell receptor gamma, IgH, and Ig kappa, right? Those are the basics that everybody is going to offer on their test. And any type of add-ons you want to do, that's on you to do individually. Penn actually took the opportunity to do some of our own personal add-ons based on feedback that we had from our clinical teams, as well as what we had previously offered. And so we took our total from the 497 up to 502 genes that we cover. You can see our gene lists here. I've included the link for you if you're interested. And I will just tell you that although we put the core set on the panel, plus the additional extras, we actually bioinformatically mask many of those genes, depending on what the clinician has ordered. And we will only tell you about a couple of them, or maybe a few hundred that you asked about. So that's an option which allows us to participate in the core set, yet be efficient with our usage and report out exactly what only everybody needs. Again, although we did participate in the group design and many group efforts, once we had our validation plan and experiments laid out, those of course had to be done in each laboratory individually. Even though we were doing the exact same experiments, everyone must do them on their own. So in conclusion today, I just wanted to review some of the things that we talked about today, and that when you're deciding to bring an assay from research and translate it into clinical care, there are so many things that you have to think about, including the clinical utility, the economic or cost of this assay, as well as if it is a priority of your laboratory or your institution. We have a huge list in our laboratory of assays we want to develop, and we keep having to rearrange priority based on our resources. So that's something you have to take into consideration. Whether you're validating or verifying an assay, there are many different parameters that you have to look through. And I provided you a chart with the minimum ones, as well as some more advanced parameters that you may need to look at if your assay has some of those are applicable to it. In the US, and I'm sure is the same in the other countries where all of you are from, there are some regulatory agencies that really govern how we can implement an assay in the clinical laboratory and exactly what rules we need to follow in order to do so. And lastly, I hope it's been really beneficial for you to hear about our experience participating with the GOAL labs and how designing an assay and doing a group group design and individual validation can offer so many benefits to all of us. We are certainly active participants with the GOAL labs. Like I mentioned, it's been started by our former trainees. And if that was a contact that anybody here wanted to have, I could certainly help facilitate some introductions with the organization that, again, helped us do a cooperative validation. So with that, here are the list of references as promised, so everyone can take those back. Some of them are very next generation sequencing oriented, so I hope that they will be helpful. And this is us. It's in downtown Philadelphia, our Penn campus. We've got some beautiful new buildings and some very old buildings downtown here. We're over in West Philadelphia. So thank you from Philadelphia. And I know that there will be some time for questions, whether it's now or during our roundtable. Thank you.
Thank you so much, Jackie, for this very interesting presentation and with lots of information that actually might help assay validation. I would suggest that we ask actually a couple of questions specific to your presentation, and then we move on with further questions on the panel discussion where we can have an opportunity for exchanging together. So I'd ask the audience if there is anyone that has a question for Jackie. Yeah, I see Felix has his hand up.
Hi, yeah, that was fascinating, especially seeing how you guys did that final, your project. But if I understand, so working with GOAL, you actually had no industrial partner in this whole process, and what are the advantages and disadvantages of that? And
well, the GOAL organization itself does have some industry sponsors to help fund everything that they do. But for us, basically going to these meetings or even meeting virtually with our colleagues in the lack of an industry sponsor, just let us all work together on the same design. And by coming to, so our probes are purchased from a company called IDT. And by coming to them with such an enormous order, as you can imagine, for probes for 29 sites with continued contracts and things, we were able to take advantage of some industry collaboration in that way by getting, some discounted pricing there. So, yeah, there definitely are some advantages. If you have the industry sponsors, there may be certain stipulations and things, and then regulating how you can spend funds, etc. So, it was really a grassroots effort of assay design. And all of us meeting just like this to come up with the best panel possible.
That's great. Thank you, Jackie. And is there any further question? We'll take a couple of questions. So if not, I'll make a very preliminary question. You talked about validation and verification of assays. Do you mind if you give a brief overview of what the differences are and why do you use these two different regulatory terms in the assay laboratory, if that can be kind of
Yeah, absolutely. The verification is going to apply to only assays in the US that are FDA approved or cleared. It is going to allow the laboratory to do a more simple process of bringing that assay up. So it has many advantages that you can purchase it off the shelf, do perhaps some quicker experiments, and you are ready to go with this FDA approved assay. There are also situations where perhaps one of them serves as what is known as a companion diagnostic, and the industry, they may be able to get better reimbursement for those in terms of insurance carriers. So there are many advantages to going with an FDA approved or cleared assay and a verification. However, the FDA does not have that many assays approved or cleared for you to pick from. And hence you need to turn to a validation, which is going to be applicable to a laboratory developed test where you design and implement it start to finish. Many advantages there are that you get to control what's in your assay, how it's done. And although a disadvantage is you're doing those extra additional steps of a validation, again, you really get to have a lot more control than if you're running an off-the-shelf FDA verified product.
Great. Thank you so much for that. I'll ask you last kindness, if you can put in the chat the link to the template that you were mentioning. I tried to find it online, but somehow these templates are buried in a beautiful website, and so that would be nice. That
absolutely, yeah, I will put our link. And you're referring to the an external test pages. Request form. Yep. Absolutely, will do.
That'd be great. Thank you so much. Then with this, I thank you again for your presentation, and we are ready for Professor Davi.